new indigenous technology is called BIRSA 101. There are many types of anemia, many different reasons for anemia. This sickle gets its name from a farm instrument, and that farm instrument is the sickle. Disturbing parts of sickle cell anemia or patients who have this sickle disease, CRISPR is a very important technology. From the point of view of UPSC exam, also already 3 questions have been asked on this. Outside that common bloodline, then the defective genes over a period of generations, they can be eliminated. Hello everyone. Today we are going to talk about some interesting developments in the field of gene therapy. So what has happened here in India is, India has launched the first indigenous CRISPR-based gene therapy for sickle cell disease. And this new indigenous technology is called BIRSA 101. So there's a lot to learn here. We have to learn about what is CRISPR, what's gene therapy, sickle cell disease, what exactly is that, and what's the significance of this whole thing, that we've developed indigenous technology on this. So what is the importance of that happening? So we'll understand things one by one. We are going to start by talking about the sickle cell disease. Okay, let's see what is sickle cell anemia. We have to learn a little bit about blood before we do that. So there's one molecule in blood, which I'm sure everybody is familiar about, everybody's heard about. That is hemoglobin. So hemoglobin is present in the blood, specifically in your red blood cells. So we have red blood cells, or RBCs as we call them, and they are containing hemoglobin. To understand the importance of hemoglobin, you should know what it does. So what this hemoglobin does is, its main role in the body is that it is to transport oxygen. Oxygen transportation is its main function. Blood is circulating all over the body. All over the body, in that blood you have red blood cells, and red blood cells have hemoglobin, which is carrying oxygen. So that's how oxygen is being delivered to each and every part, each and every cell of your body. Now what this oxygen does, oxygen's purpose is production of energy. To produce energy, the reaction through which energy is produced in the body, that is done with the help of oxygen. So here you can understand the whole flow of things. So if you have, let's say, less hemoglobin, you're going to have less oxygen circulating in the body, and that means you're going to have less energy, which would make a person very weak. And this situation where you have less hemoglobin, you have a technical term for that. It's called anemia. So when you have less hemoglobin, that is called anemia, right? Now you would have heard about anemia. There are many types of anemia, many different reasons for anemia, but here we are going to talk about one specific reason for anemia, which is linked to the structure of red blood cells, the shape of red blood cells. Let's just look at this and try and understand. This is your normal red blood cells. The red blood cells, they are normally in this disc-like shape, and this shape is ideal to store a lot of hemoglobin. But sometimes, under some special conditions, some defects basically, this disc-like shape can change into this sickle-like shape. This is a deformed and improperly formed red blood cells. This sickle, if you just look at this shape, this sickle gets its name from a farm instrument, and that farm instrument is the sickle, because the shape is like that. These are called sickle-shaped red blood cells. Now, the problem with having these sickle-shaped red blood cells is, first, that they'll have less of that normal hemoglobin. So the shape itself is like that, that there is the normal hemoglobin levels will become less, because in this whatever is coming, whatever is getting accommodated, there that is going to be abnormal. So the first problem is that you have less normal hemoglobin, okay, which means anemia, which means the person is tired, there is weakness of the body, right? Another problem which happens with this shape is, see, red blood cells are flowing through blood vessels. And in the blood vessels, when these abnormal shaped red blood cells are flowing, there is a possibility that they can get stuck. So they are stuck, they obstruct the blood flow because of that. They're going to be stuck in that blood vessels, in the smaller blood vessels like capillaries. So this shape finds it's difficult to move around in that. So wherever they get stuck, there is an abnormal blood flow there, and that leads to a lot of pain. So one of the most disturbing parts of sickle cell anemia or patients who have this sickle disease is that they experience a lot of pain in different parts of the body. And because of less hemoglobin, less energy in terms of the kind of life they'll lead, it's not going to be a very long life, because you need healthy blood for normal existence. And a lot of people, a lot of children who have this disease, they are sometimes not even able to live more than 5 years of age. So because of that, this problem is a very, very serious problem. Okay. And the bigger tragedy of this is that the treatments which we have for this, those treatments are not very good, not very effective. If we look at how we are going to treat this, okay, we don't have to go into specific aspects of treatment in terms of medicine or anything like that, but just I'll give you an overall sense about what is being done right now. So one thing is you can give blood transfusion. If the person is becoming very anemic, blood is very low, you can give new blood. Blood transfusion can be given, but that's not really a good or a long-term treatment, because red blood cells have a life of 120 days. So even if you're giving new blood, that blood will need to be given again after 120 days. Okay. Another thing is you have some drugs, some medicines which are there, which increase uptake of oxygen. Like whatever functional hemoglobin is there, you can make that hemoglobin itself work in a way that it can take up and it can transport more oxygen. Okay. But again, not like a foolproof treatment or a cure. But yes, this is one of the most effective things as of now, which is done, which is given to maintain these patients. And along with that, you have to also keep monitoring them. Other than this, there are two more options, which are the technically advanced options, where you change the red blood cell formation system. One is that you change the bone marrow of a person. Now, the sense in which bone marrow is significant here is like these red blood cells. In fact, all the blood cells of the body, they are formed in a special location called bone marrow. Bone marrow is a soft tissue material which is found in the long bones of the body. Long bones means bones of hands and feet. So in these bones, there is a soft material called bone marrow where all the red blood cells are formed. Now, in sickle cell anemia, we say that the red blood cells which are formed are defective. So what we can do is that we can change the bone marrow itself. You might have heard about bone marrow transplant, that you take somebody else's bone marrow who is healthy, you give it to this person, and then the person will start producing normal bone marrow cell. But in this, there are complications of matching right? Bone marrow should match. Sometimes it matches between family members, sometimes it doesn't. So you have to take it from somebody else. So that whole process of matching and transfer, that is tricky. We have now formed or created a new technology which uses the person's own bone marrow but with slight modifications. And this is where gene therapy comes into place. So, why gene therapy, or what exactly will happen here? To understand this, we have to understand the reason for sickle cell anemia happening. That why is this defective sickle cell being created? Okay. Now, for all the properties which we have, for all the traits which we have, there are genes present for all our characters, like what kind of hair I'll have, what kind of eyes I'll have, how my liver will work, how my pancreas will work, how my red blood cells are formed, that is all decided by genes. So these genes, they are segments of DNA. So basically it's DNA, right? I'm sure you've heard about DNA and you know everything is decided by DNA. From head to toe, everything is determined by DNA. So here, segments of those, that DNA is large, DNA is a very huge molecule, and these have different genes for different purpose, like gene for color of hair, gene for producing insulin in the body, gene for deciding hemoglobin, gene for deciding red blood cells. Now, there are more than 20,000 genes in human beings. Okay, human beings have more than 20,000 genes for all the different characters which are present. In sickle cell anemia, so sickle cell anemia, if I mention here, sickle cell anemia, or you can call it sickle cell disease, in this, there is one gene which is defective. There's one gene which is not working properly, and because of that gene not working properly, the shape of red blood cells are deformed, the hemoglobin formed is deformed, and the person has anemia, the person has abnormal red blood cells. So in gene therapy, the focus is going to be on changing that gene. There is a gene which is deciding all of this, right? And which is deciding the shape. So if I can find a way to change this gene itself, then my problem is going to be solved. Okay, my problem of how the disease is being transmitted, that is going to be solved if I've corrected the gene itself. Now, there are many different ways in which this can be done. We are going to talk about the most successful technology, the most successful gene therapy, which is CRISPR, and we'll see how it can be used in sickle cell anemia. Then we'll come into the specific context of the disease in India and what is the repercussions of this whole thing. Let me first give you the basic fundamentals of the gene therapy, and the specific gene therapy which we are using here is gene editing. I'll bring you to the specifics here. Just go through this with me and try and understand every part here. So gene editing as a term basically means it's a process in which you can add, delete, or replace genes. You have developed this technology that whatever genes are there, any gene, you can add an extra gene. You can, like sometimes a gene is missing, or if it's defective, you can replace it. If it is deleted, you can add it. All these possibilities are there. What you have to do in this is that you take a cell, genetic material or DNA of a person is present in the nucleus of the cell. So nucleus of all cells, of all living organisms, they have genetic material, and this is present in the form of chromosomes. They are rod-like structures. Basically, this is the shape in which DNA is going to be present. Now, let's say this is that section of DNA, and this red color thing is the defective part which you want to edit. I told you there are 20,000 genes, right? So, out of these 20,000 genes, the most difficult task is to identify that one gene. Like in sickle cell anemia, if you have to identify the gene for hemoglobin formation, okay, or the shape of red blood cells, then you have to reach that exact location. You have to precisely go at that point, and for that, you can make a specific guide molecule. This is a very important part in gene editing, that you need something to guide the system to that location. Whatever editing tool you are using, it has to reach that location and only that location, right? You cannot edit at a wrong place, because then that is going to serve no purpose. So there's a guide molecule, and along with that you need something to cut at that site. So there are specific enzymes which do this function of cutting, and this DNA cutting enzyme, it is going to travel with the guide molecule, so that the guide molecule can take it exactly to that precise location. So there's a guide molecule, there's a DNA cutting enzyme. Now that you have cut at that specific site where you had, let's say, the defective DNA strand, now all opportunities are open. You can add a gene, you can delete it, you can let the cell repair it into a healthy strand, you can replace the gene, whatever you want, that can be done. But the two important steps here are guidance and cutting. Okay. The specific technology which we are talking about here today is CRISPR. So let's see how this whole thing specifically happens with CRISPR. CRISPR is a very important technology. From the point of view of UPSC exam, also already 3 questions have been asked on this. It has been given the Nobel Prize also. It is a very revolutionary, very significant, and very effective technology. Okay, so we need to know about this. The full form of CRISPR is clustered, regularly interspaced, short palindromic repeats. Okay, or in short, CRISPR. Now, CRISPR is your gene editing tool. The first thing you need to know about CRISPR is that CRISPR is not something which human beings have invented or created. CRISPR is a natural mechanism which bacteria use to protect itself. It's basically a part of bacteria's immune system. Okay. We have learned from there, and we've used it to create a technology. The technology will have two parts: guide molecule and DNA cutting enzyme, as I have just now discussed with you. I'll tell you here the specific names of these. So, the guide molecule is a special type of RNA. Like DNA, there is another kind of genetic material which does different kinds of function, that's called RNA. And in this also, a specific purpose RNA is there, which is called guided RNA. Okay, sometimes you'll find that because there can be different types, it's also written as single guided RNA. It means one of the same thing. Basically, what you need to remember is that there's an RNA involved for that guidance purpose, taking CRISPR to that specific location where you want to edit. And then you have a DNA cutting enzyme, which is called Cas9. So Cas9 protein, basically all enzymes in their chemical nature, they are proteins. So this is called Cas9 protein. This is the cutting enzyme. This will cut DNA. This is also called molecular scissors. This was one UPSC question, that Cas9, what is it? The answer was molecular scissors. Okay. Now this CRISPR is being tried globally. This technology has been around for many years. The first disease where this is being tried is sickle cell anemia, and few people have been treated of sickle cell anemia using CRISPR. Okay. The problem with those kind of treatments is that it's a very expensive treatment. Okay. The procedure is long term. It takes weeks or months to treat a person. And in sickle cell anemia treatment, basically what you are going to be doing is, like, you have these bone marrow cells which are producing red blood cells. But the person who has sickle cell anemia, his bone marrow has defective red blood cells. Now, this is what you are going to change here. In your gene editing, what you do is, so there is a person who has sickle cell anemia. You take this person's bone marrow, the bone marrow which contains cells which will make new red blood cells, but this bone marrow is defective, right? That's why it's making defective cells. So there is a defective gene in its cells, right? You take this bone marrow out, and after that, what you're going to do is you're going to do gene editing on this using CRISPR. And using this technology, what you will do is the defective gene is going to be changed. And once this editing is done, the cells will no longer have the defective part. So the bone marrow now will be able to produce normal red blood cells. This you're going to put it back into the person. So this is the person's same person's bone marrow. You take it out, you do gene editing, you send this normal bone marrow back. Now this normal bone marrow will produce normal red blood cells, and the person's sickle cell disease is going to be corrected. The cost of this treatment, because it's very early on, the cost is in millions, like 2 million, 3 million, that kind of, uh, sometimes even more. That's the kind of cost which goes into these kind of treatments. So India, what it is doing here is that India is trying to create its own technology, its indigenous technology, so that it can be made affordable. We don't have that kind of millions of rupees to spend on the treatment of diseases. So the idea is that creating our own technology. See, one way of using this technology is that, okay, somebody is doing this in USA, you get a license from them and you do that manufacturing there. But that's going to be expensive. So if you can create this technology on your own, then you can obviously control the cost. So that's the significant thing which is happening. And I'll tell you how incidence of this disease or status of this disease is there in India, and why this is so essential that the cost be monitored. So in India, there is kind of a disproportionate existence of this disease in terms of all the world statistics. What's happening is that the number of people suffering from sickle cell anemia in India, it's very high in tribal groups. Tribal communities have a very high proportionate, like they say that 1 in 86 people die because of sickle cell anemia in certain tribal groups of central India, eastern India, places like Madhya Pradesh, Jharkhand, Chhattisgarh, even like parts of Rajasthan. They have a very high incidence of sickle cell anemia. Because what happens with sickle cell anemia or anything which is a genetic defect, we all have genes, we've talked about that, we have DNA, but how we are getting this is we are getting it from our parents. Okay. And they are getting it from their parents. So there is that continuous line of inheritance which happens with genetic material. Okay. And when you are marrying within smaller groups, like this concept of endogamy, if you understand this word, endogamy. Endogamy means when you are marrying within close relatives and having children within those small blood related groups, the chances of genetic diseases are very, very high. Like, think about this. There is a person who has sickle cell anemia. All his children will be getting those defective genes. Then their children will also be getting the defective genes. Then their children will also be getting the defective gene, and the disease will keep continuing in their family, as opposed to a situation where maybe if they were marrying outside their community, outside that common bloodline, then the defective genes over a period of generations, they can be eliminated. But in endogamy, that does not happen. And for tribals, I'm sure you understand this. Tribals, like let's say there's a tribal community, let's say they have 5,000 people. So their marriages, their children are coming from amongst those 500 people only, right? So the disease, the defects will keep continuing. That's why they have a higher incidence of the defective genes and hence sickle cell anemia. So the targeting or the treatment of sickle cell anemia is for tribal groups. They as such don't have access to very good healthcare facilities or modern equipment or financial resources. So the cost of the treatment, whatever treatment you want to try, that is very, very significant. It has to be affordable. That's the only way you can avoid these diseases. The government is talking a great deal about this. There is a special dedicated mission, which is National Sickle Cell Anemia Elimination Mission. Okay. This mission is set with the target that we have to reduce the cases, and because it's mostly in the tribal groups, the treatment, the mission is targeted towards these tribal groups. Okay. As of now, the significant thing which we are doing is, or which is a part of this mission, is that you do early genetic testing. Test people for genetic diseases for sickle cell anemia so that they are aware. And it should also be a part of marital counseling also, that if people have defective genes, they should be discouraged from having children or not marrying the person of that same group of defective genes, right? So that is being done, but the short treatment would come through gene therapy. As of now, it started happening globally, but in India, we haven't started using it. We've just started designing our own indigenous technology. And with CRISPR, we'll be able to save a lot of lives lost and a lot of people which live a very miserable life because of sickle cell anemia, because of sheer amount of excessive pain and weakness and debilitated condition. Now let's see what is this India's technology. So this gene therapy can be of in many different ways. We are using the specific gene editing CRISPR here. It's called BIRSA 101 after Bhagwan Birsa Munda, the great tribal freedom fighter. So I told you the disease is more common in tribal communities. So the name of the technology is also taken from a tribal idol, which is Birsa Munda. So that's why it's called BIRSA 101. Now, this technology, the indigenous CRISPR technology for sickle cell anemia, it's being developed by CSIR and its Institute of Genomics and Integrative Biology. So they have created this technology and now they're doing a technology transfer to a startup, which is Serum Institute of India. Serum Institute of India, you would have heard about it. It's been very instrumental in producing vaccines, especially during the COVID time. It became very prominent for doing this. So the reason why they're doing a technology transfer here is because they are the ones who will do the manufacturing, right? Somebody has to manufacture and sell it also. So the idea of doing everything in-house, that okay, technology was designed and then manufacturing will happen in India only through an Indian company, is to control the cost and to be able to give that treatment to a lot of people who are suffering from this disease. Okay, so the technology transfer process has started. They'll start manufacturing and then we'll have this available for treatment. Okay, this would really cure people of sickle cell anemia, which otherwise has no cure. So this is the significance of it, why India is doing it on its own to reduce the cost and to target its people. You have a very large number of people in those tribal groups suffering from this. They say at least there are communities in which 40% of the people are carrying a defective gene in those tribal communities. So this is a very significant thing for India and a very big achievement for the Indian scientists to have brought this BIRSA 101. With this, we conclude the discussion for today. Thank you.